that are operating inside you right this very second. We are, of course, talking about cells. I mean, just look at that number, 100 trillion. That's a rough estimate of how many of these fundamental, microscopic units actually make up the human body. Every single part of you is constructed from trillions of these individual living blocks. And yet, there's no way you can see a single one of them with the naked eye. They're literally the invisible foundation of our entire existence. So here's our game plan for today. Number 1, the invisible world. 2, boundaries of life. 3, the cellular factory. 4, power and storage. And 5, growth and cell theory. All right, let's jump right in. Kicking things off with Section 1, the invisible world, seeing the unseen. To really understand the cell, we kind of have to understand how we overcame our own biology to even see them in the first place. The human eye has a strict limit of resolution. It's about 0.1 millimeters. If 2 points are closer together than that, they just blur into one single blob. And cells? Well, they are way, way smaller than 0.1 millimeters. That's exactly why we need powerful tools like light microscopes and electron microscopes just to see them. This whole journey actually started way back in 1665, when a scientist named Robert Hooke looked at a slice of cork, saw these tiny little box-like compartments, and called them cells. Moving right along to Section 2, boundaries of life, crossing the threshold. Now that we've got the tech to see them and we've zoomed in, the next big question is, how do things actually get inside the cell? This everyday potato experiment brilliantly illustrates how that outer boundary, the cell membrane, works. It's a selective barrier. Think about it. If you put a piece of potato in plain water, it swells up. But if you drop it into a 20% salt solution, it shrinks. Why is that? Well, it's because the cell membrane allows water to pass through, but it blocks other substances. It's constantly reacting to whatever is going on in its environment. So the crucial concept here is osmosis. That's the scientific term for the potato shrinking and swelling we just talked about. Water is moving across the cell membrane, which, by the way, is this incredibly thin, fluid double layer of fats, trying to balance out the concentration on both sides. The membrane's proteins basically act like tiny bouncers at a club, deciding what gets in and what gets kept out. But, not all boundaries are created equal. Just contrast a plant cell with an animal cell. Plants can't exactly run away from a storm, right? So they need a really rigid outer structure to withstand wind and rain. That's their thick cellulose cell wall. Us animals, on the other hand, we need our cells to be flexible so we can actually move around. So our cells skip that rigid wall completely. Let's zoom out for just a second to look at the 2 main types of cellular architecture. First, we've got prokaryotic cells, like bacteria. These are primitive, they're super small, and their genetic material is just kind of floating freely in a nucleoid. Then we have eukaryotic cells, like ours. These are much larger, highly complex, and they store all their genetic instructions inside a super secure, membrane-bound vault called a nucleus. And that brings us perfectly to Section 3, the cellular factory inside the organelles. We are now breaching the inner, jelly-like cytoplasm of a eukaryotic cell to take a fast-paced tour of the factory floor. Right smack in the middle of everything is the nucleus. Think of this as the ultimate control center, or like the executive boardroom. Inside, safely locked away, are chromosomes made of DNA. Those are the literal blueprints for everything the cell is and everything it ever does. So the nucleus is the house of coded instructions. But actually, here's a totally wild exception to that rule. Mature human red blood cells don't even have a nucleus. They literally eject it to make more room to carry hemoglobin and oxygen around your body. But because they have no instruction manual to repair themselves, it limits their lifespan to only about 120 days. Crazy, right? Next up, we follow those instructions from the nucleus outwards into this massive folded network, the endoplasmic reticulum, or ER for short, and its partner, the Golgi apparatus. This is the heavy industry sector. The rough ER is studded with these tiny structures called ribosomes, which makes it look all bumpy under a microscope. These ribosomes are the workers on the floor, actually manufacturing the proteins. Once they're built, the proteins get sent to the Golgi apparatus, which acts exactly like a post office. It modifies them, packages them up into little vesicles, and ships them off to exactly where they need to go. But what about the waste? I mean, every factory produces trash, right? That's where lysosomes step in. They are the ultimate cleanup system. Basically sacks filled with incredibly potent enzymes that break down worn-out cell parts and fight off foreign invaders. And get this, human sperm cells use these exact same lysosomal enzymes in their heads to break through the outer layer of an egg cell during fertilization. All right, moving even deeper, we hit section 4, power and storage, fueling the cell. Let me introduce you to the mitochondria. Structurally, this thing is just brilliant. Notice that unique, heavily folded inner membrane? Those folds are called crista, and they exist to maximize the surface area inside the organelle. And in the cell world, more surface area means more space for vital chemical reactions. Those reactions are exactly why it's famously known as the powerhouse of the cell. The mitochondria take glucose and other molecules and break them down during a process called cellular respiration. This releases energy, which gets stored as a tiny molecule called ATP. Think of ATP as the primary currency that fuels literally every single cellular activity in your body. But hey, you also need a place to store your supplies. Enter the vacuoles. In our animal cells, they're pretty small and temporary. But in plant cells, there's typically one massive central vacuole filled with water and nutrients. It acts kind of like a giant internal water balloon. When it's full, it pushes against that rigid cell wall we talked about earlier, keeping the whole plant firm and upright. But when it loses water, the pressure drops and the plant wilts. Finally, let's zoom back out for section 5, growth and cell theory, multiplying the workforce. To sustain life, these microscopic factories can't just operate. They have to multiply to continually build and repair the body. This happens through mitosis. It's like a highly choreographed dance. A single parent cell prepares itself, precisely duplicates its DNA blueprint, and splits right down the middle to form 2 genetically identical daughter cells. This exact process is happening in you right now to heal your skin if you get a paper cut. It just continually replaces lost cells. And we know this isn't just some abstract theory. Look at this microscopic view of an onion root tip. Because a root is constantly growing, you can clearly catch the cells in the act, showing off the different stages of cell division. It's a very real, visible, mechanical cycle taking away endlessly to produce growth. Which brings our incredible spatial journey full circle to the unifying principle of all biology, classical cell theory. Formulated by scientists like Schleiden, Schwann, and Virchow, it simply states, all living things are made of cells. The cell is the basic unit of life, and every single cell comes from a pre-existing cell. It is literally an unbroken chain of life. But I want to leave you with one final, kind of wild thought. Normal animal cells eventually stop dividing when they bump into neighboring cells. It's a mechanism called contact inhibition. It's a beautiful, elegant balance. But what happens when that balance breaks? When the factory loses the instructions and forgets how to stop building? Well, the cell divides uncontrollably. And that, unfortunately, is the origin of cancer and tumors. It is a stark reminder that life at the microscopic level is a delicate, precise, and utterly vital balancing act. Thanks so much for joining me on this explainer, and keep exploring.